and welcome back to another video of Smarter with Lana. In this video, I'm going to be telling you about messenger RNA vaccines. So let's start by talking about traditional vaccines. What are vaccines? Vaccines mostly include dead or weakened pathogens that would trigger an immune response involving adaptive immunity in our body. When these dead or weakened pathogens are injected into our body, cells from the adaptive immune system will be able to produce antibodies that could block these pathogens from attaching to our cells and causing harm. Memory cells, on the other hand, will make sure to remember what this pathogen looked like for future encounters, so that in future encounters it will be able to recognize it straight away and produce the needed and specific antibodies for it. So hopefully this will make you realize that the symptoms you get when you take a vaccine are not directly caused by the pathogen, but they are caused by your immune system trying to combat and fight off that pathogen. Fever, inflammation, all of these processes are aiding your body in getting rid of the pathogen. However, keep in mind that at the same time, inflammation and fever are not good for your body. What are mRNA vaccines or messenger RNA vaccines? Messenger RNA, from its name, think of it as that dude who carries information from your DNA to the factory where all the proteins are made. So again, messenger RNA takes information from your DNA in the nucleus and sends it to the ribosomes, which are the factories where your proteins are going to be made. Now these ribosomes or these factories will be able to translate what the messenger RNA is saying, and then they'll be able to exactly know what the DNA was saying based on what the messenger RNA was carrying. So let's go back to vaccines. Instead of us actually producing the pathogen, actually producing these weakened or dead pathogens and then injecting them into the body, what if we can tell our body to make that for us? Obviously that will save a lot of time and a lot of money, and this is especially helpful when you're producing a vaccine for a pandemic, something that's going to be used hopefully all around the world. Reducing the cost is a really good thing. So what scientists did is that they figured out the DNA sequence of the spike protein on the outside layer of the coronavirus. So this is the coronavirus. You'll see that there are these small protrusions on the outer surface of the virus. These are the spike proteins, and these are the things that allow the virus to attach to our cells. The clever thing is that when they figured out the DNA sequence for that spike protein, then they were able to create the messenger RNA sequence of it and inject it into our bodies. So this way, our body will be able to detect these specific spike proteins that are on the surface of the coronavirus or the novel coronavirus. So when that messenger RNA vaccine is injected into our bodies, that messenger RNA is actually going into the cells, and the cells, the ribosomes inside your cell, are making that spike protein, which will then be encountered by immune cells from the adaptive immune system. Now, if you remember, when these immune cells encounter a pathogen, and here it's going to be for the first time, they will then learn how to produce antibodies that are specifically made to fit that pathogen. So let's put it simply. By injecting the recipe that is needed to make the spike protein on the outer shell of the coronavirus, we are enabling our bodies to get to know this spike protein without the actual virus, without the genetic material found inside the virus. We only have the spike protein. We are enabling our cells, we are enabling our immune cells to get to know that, just in case they encounter the actual virus in the future. Now, unfortunately, messenger RNA vaccines, they do save a lot of time and a lot of money, but the problem is that that messenger RNA is a foreign substance that is being injected to your body, so you'd assume that your body will fight it off. And it does. That's why the production of messenger RNA vaccines is not easy. Another thing which was really difficult for scientists to get it sorted was the stability of that spike protein when it's detached from the actual virus. Proteins have 3D structures, and these 3D structures give the protein its stability and its function. These 3D structures are also influenced by other proteins around it. So the fact that we've detached that spike protein from the actual virus does reduce the stability of the spike protein, which created a massive backdraw for the scientists who were working on it, but we finally have the vaccine. Thank you so much for watching. I hope that you know a little bit more about what messenger RNA vaccines are. If you like this video, like, subscribe, and all the YouTube stuff that you know about, and I'll see you next time. Ciao!